Okay. So I will be honest and say I think the light reactions are the more complicated of the two, as you can probably tell by this diagram. Um, I just need you to have a basic understanding of what's going on. You don't have to have it memorized by any means. You would never be asked to label it. But you should just have it like, what the heck is the point of the water? Like, what does the water do? And how do we actually make the ATP? So that's my goal, for get you to know. So within the light reactions, and within the chloroplasts and the chlorophyll themselves, there are things called photosystems. And notice there are two of them in this diagram. There's something called photosystem 2 and then photosystem 1. And I know like numerically they're out of order. There's a reason for that, but it's not important. So I won't even share it with you. It's a really dumb reason. But if you ever want to know, feel free to ask me. So basically, a photosystem is a different collection of pigments. Different pigments that are there, different types of chlorophyll in different amounts. And they focus on slightly different wavelengths. So this one is really focused on 680 nanometers. This one's focused on 700. So they just absorb slightly different ones. So what happens in the photosystem is it's just a collection of all these pigments in like one general area. The photons of light shine on the chlorophyll. And the yellow arrows that you see here are representing electrons. The electrons are bouncing around to different chlorophyll molecules. And then they hit this one, it's called the final electron acceptor, that boosts the energy level. Oops, I wanted to show you. Notice the little electron here. So what we're doing is we're exciting electrons, and we're also going to steal some electrons from water. So water, of course, by the way, is being absorbed through the roots of the plant. But when it gets to the chlorophyll, it actually breaks apart. We want the hydrogens. And then the oxygen, which you see down here, I know it says 1/2 of an O2, and that's because O is by itself, and then you wait for two of them to combine, and then you get O2. So anyway, oxygen is just released like junk, and we want to keep the hydrogens. But we separate the hydrogen proton from the hydrogen electron. So that's why you see this lone little electron up here and the proton down there. And we're going to keep that proton on this side. So the electron gets really excited, and then it goes through this right here, this little chain, the slide. In short, this is called the electron transport chain, or the ETC. That's not really super important. It's also labeled at the bottom here, electron transport chain. It's a series of proteins. Okay, so the electron transport chain is a series of proteins in which the electrons pass through. Okay, and as they pass through them, their energy levels drop, which is why it's a slide going down. Then it hits another set of chlorophyll molecules called photosystem 1. And in photosystem 1, you've got the light shining down on the chlorophyll again, exciting and boosting electrons, raising electron energy levels here. And then this electron, where it's going to go, is going to eventually end up on a molecule called NADP+. And then you're going to add in that electron and a random hydrogen and form NADPH. So basically, the NADPH's job is to carry those electrons that we had originally separated from their protons. Okay, so that's going on when light shines on plants. It's a lot. The main part, and the reason why we talk about this light-dependent and photosystem business, is this word at the very bottom here called chemiosmosis. And yes, that is the word osmosis. Chemiosmosis is going to help the cell make the ATP. So there's one more step that's going to happen. How do we actually make the ATP from this hot mess of a reaction? Okay, so let me just review what we said. The electrons that we're going to be moving in this electron transport and hiring the energy levels, it comes from splitting up water. We discard the oxygen gas as a waste product. And by the way, how does the gas get out? It gets out through the leaves of the cell, and on the underside of the leaves are little openings called stomata. So that's where the O2 goes out. It's also where the CO2 comes in. Okay, so anyway, we get rid of the O2. We're going to start to accumulate hydrogen protons on one side, creating a gradient. So we're going to get a high number on one side and a low number on the other. And if you know anything, if you get a high on one side and a low on the other, you have a bunch of potential energy for movement. And that's what's going to drive the formation of the ATP. The electrons that we've stolen from the water are going to move through a series of proteins called the electron transport chain. The purpose of moving the electrons is to help create that hydrogen proton gradient. The electrons are eventually removed from the process by NADP+, and they form what's called NADPH. So it's just a way to remove the electrons when you're done using them. Okay, so then there is a molecule called ATP synthase, and that is, okay, so first of all, notice the ASE ending. That means that it is a protein. It's a very special protein that's embedded in the cell membrane, not the cell membrane, the chloroplast membrane, whose job it is to actually phosphorylate and make the ATP molecule. The way it works is we've got all this potential energy from having all those hydrogen protons accumulated on one side, and they are going to flow through the ATP synthase from high to low concentration. So this getting back to this word called chemiosmosis. So chemiosmosis describes the movement of these protons from high to low. Chemi just means chemical, and osmosis is kind of a play on words. In this case, I know you think it moves, well, I taught you correctly, I think, that it means to move water from high to low, but we're sticking the word chemi in it just to say, oh, we really just want to move chemicals from high to low. Now, there's a really good video here, and there's a better link to it. You don't have to obviously type that in. That would be a mess. But there's a link to it at the end of this little slide presentation I'll show you that does everything I just said and walks you through it probably in a more coherent way with pictures, so showing it to you animated. I highly recommend watching that. You don't have to memorize everything because they're probably never going to ask you the details, but you have to understand chemiosmosis. I feel like that pops up on the exam every single year. So I included one more picture of it here just to kind of review some of the ideas. Let's do red. Yes. Okay. So this is the thylakoid membrane. I mean, they're not showing it to you really well here. Outside the cell, or outside the thylakoid, inside. You got light coming down, shining on a photosystem. They've withdrawn the details just to make it easy. He's showing you water going in. We're going to get rid of the O2, don't care about it. We're going to accumulate these hydrogen protons, and then the electron we stole from the hydrogen is going to move through the electron transport chain. As it's moving through these chains, notice that's helping to move more hydrogen onto this side and creating an even greater gradient. Okay, then the electron arrives at the second photosystem where its energy level gets boosted again, and then once that boost happens, party's over for the electron, it finds an NADP molecule and becomes NADPH, and then it's going to be used again later, but for now we just move around. Okay, so now you got all these hydrogens on one side and very few on the other side. This is the new star of our story. This is the ATP synthase protein. And it is sole purpose is to allow the hydrogens to move across. As they move across, it actually spins the ATP synthase molecule, and that motion allows the phosphorylation of ATP. So it adds the P back into the ADP to form ATP. So the big deal here is that we've made an ATP molecule. So that's really what chemiosmosis is all about. There's another link to a video at the bottom here that can help show that to you in an animated way. Okay, so that's the end of the light. So that was just about light. That's what's happening in the daylight hours here. So just some highlights. The whole purpose here is to store up ATP because we're going to use it in the next step to build the sugar. What we need in this process is light and water. What we produce is ATP, NADPase, and then remember, we're saying it produces oxygen, but the oxygen is really just a waste product. Really good trivia question, though, and one that has been on the AP exams in the past is, the oxygen that you are breathing right now, where did it come from? Where did it originate? And the answer is water. It used to be part of a water molecule, not carbon dioxide. So it's not like a gas-to-gas conversion. It's actually water vapor, liquid, into oxygen gas. So that's kind of interesting, too. Okay, now the good news is, there's not a whole lot to know about step 2. Okay, so step 2, about the light independent. Remember, these are the things that happen in daytime or at nighttime. They are synonymous with the Kelvin cycle, although you don't have to know that. And we are no longer in the thylakoid. We don't need to be in here anymore. We've harvested enough energy. We're storing it up. Now we need to use the energy to build the sugar, which is what you see at the bottom here. So I boxed out what the Kelvin cycle is actually like. we're going to use, I already mentioned that. I think that's all I want to tell you. So, that's one other thing. The carbon dioxide that plants absorb through their stomata, by the way, the openings in the leaves of cells, or cells of leaves, is going to be used here. So we don't even need carbon dioxide until the very end when we have to build our sugar molecule. Oh, and one more thing, pardon me for flipping back a million times here. I just want to mention the law of conservation of matter. Notice that a glucose molecule is C6H12O6, 6 carbons, C6, and then notice that it takes 6 molecules of carbon dioxide in order to make this work. So a coefficient of 6 here, 6 carbons, 6 carbons. So it actually does take 6 molecules of carbon dioxide to make it work. So everything is well and balanced. Okay, so here's the big statement. You do not need to memorize anything about the Calvin cycle. I used to know it really well, but now that they've removed it for so many years from like common knowledge, you probably only have to know the details if you took a cell biology class in college, a really detailed one. But I just want you to notice a couple things. So see the CO2 coming in. Also I want you to pay attention to the arrows, the direction of the arrows. Notice ATP going in, that means it is being used. NADPH is being used. And then more ATP is being used. So what happens is the carbon dioxide gets absorbed, broken down, and it gets like reformed a bunch of times into all these intermediate molecules. Like this one has, 6 carbons, then 3 carbons, then 2 carbons, then 5 carbons, and it goes around and around. It actually goes around 2 times. I shouldn't say that. It doesn't go on forever, but it goes around 2 times, and eventually you build, this thing at the end here, this C6H12O6. You do not need to know any of these other ones. PGAL, RUBP, not necessary. It just happens to be on the diagram. So you just need to understand the Calvin cycle is about using the carbon dioxide and all of your energy to build your glucose molecule. Okay, so we're just about done. Finally, see, the Calvin cycle was really easy. So another picture here, just summarizing light dependent versus light independent, who's involved, where are we in the cell. The big focus for you should be what goes in and what goes out, what is used and what is made during each of these 2 stages. I'd say that's the most important part.